So, hello everyone. Microbehunter here. I want to welcome you again to another Saturday microscopy live stream. Today I would like to show you a couple of prepared microscope slides about insects, and yeah, the one guy that you already see over here on my side, that's an aphid. So several slides I made myself, other slides are commercially prepared, and most of the slides I have not looked at myself yet. So it's going to be also new for me, because some time ago I bought myself a slide box with ready-made slides. A few weeks ago we already looked at a few of those slides together, but I did not look, or we did not look at the insects yet, so I would like to do that today. Yeah, I hope that everything works again, because I did receive a message the other day that there was some kind of a problem at the beginning, some delayed sound. I hope that this works now, but I see that many people are now joining as well. I would like to simply invite you to ask microscopy related questions. Please always add at the beginning an @Microbehunter or @Oliver, so it's easier for me to find the questions that are the comments that are directed to me. Otherwise, I just want to say a happy new year, of course, all around the world. I'm happy that the sound seems to work, and I'm happy that so many people are joining in again. I do also have the other page open, and I'm just seeing, there are 51 people right now in the live stream. That's quite nice, okay? I'm quite happy that so many people are interested in microscopy, especially all around the world. And I'm happy that you found some time. So what I'm gonna do is the following. I'm going to quickly show you now the desk that I have here. So that's my desk camera. And what you have over here, I've got, look, all of these are ready-made slides of insects and insect parts. And there are a couple of slides that I made myself here. You're probably going to notice that Varroa mite, that's a mite, that is not an insect. Also in ticks, they belong to, they're ticks, they're not insects, but it doesn't matter. We talk about this later, but also over here I've got some mites, some of which I already showed you before in the previous live stream, the Varroa mites, but there are plenty of other specimens over here that I did not look at yet. Butterfly of a wing, butterfly wing, dragonfly wing, honeybee wing, different types of wings, and so on. So we're going to have a look at those, and it's going to be also a little bit of a surprise to me as well. Okay, so that's a little bit the plan that I have for today. We're just going to look at those specimens here. There might be, of course, questions that I'm not able to answer, but I'm just not able to answer them in this case. Okay, I'm just always every now and then I am having a look over here always into the chat. There might be a small delay here. And will you show compound eyes? That is has been a question right now as well, and the answer is going to be yes. As a matter of fact, I'm going to do that. So, but a tiny little bit of biology I'm also going to do here. This here is an aphid, and we can already see here the compound eyes, and it's an insect. Why do I know that? Because you basically count the number of legs. So we've got one leg pair, 2 and 3, so there's 6 legs present. And aphids, look, they have this long trunk over here. Aphids are those tiny little insects that sit on plant leaves, and they basically use the little trunk here to puncture the leaf and to suck the sap of the leaf, and this can actually destroy the plant. Okay, and one of the nice things about many of those specimens is that and I also mentioned this already before, is that the insects were prepared in such a way that they became more transparent. So you see, it's not completely black what we're seeing here, but as a matter of fact, it's they're a little bit transparent. Now, in this aphid, I found something else that is kind of interesting, and I'm going to now go down with the magnification. I have to also go down here so that we see the whole insect better. Okay, here we go. So that is now, you can always look at the top here, that is the 4x objective. And I want to show you something over here. Do you see those little strange structures over here? This one over here, and this is the second one over here, right? And this one over here, and this one over here. those appear to be basically the 2 pairs of wings that now basically are not used for flying anymore, because the aphids, they don't fly around anymore, but just sit on the leaves and suck the sap. But look, that's basically whatever is left over of the wings, because that's also one characteristic is that there are 2 pairs of wings present. Now, flies only have 1 visible pair of wings. I'm going to talk about this as well. So there has been already a question about the compound eyes. So I'm going to zoom in again all the way. Need to turn up the light again here. Okay, and open this up here a little bit. It always sometimes takes a little bit of time for me to adjust everything. Yeah. And of course, very typical for insects are the so-called the compound eyes. You can see those little tiny circles here. These are the individual ommatidia. So basically, these are the individual light-sensitive elements here. So that's also very typical of insects, because spiders, they have do not have compound eyes. They actually have lens eyes. Okay, so. And there are already some questions over here, and there's some comments I also would like to read here in the comments section, the chat section. I had a great insect experience with my students this week. View mosquito larvae under the scope and discovered they had hitchhiking epistesis. It looks like vorticella over much of the bodies. So that's also something that sometimes larvae that have been in the water for quite some time, they've got other microorganisms growing on them. Okay, what is another question for me from Marcel? What is, in your opinion, the best mounting agent, the best mounting medium for insects? If you want to make a permanent, that's an easy one to answer, luckily, because there is a mounting medium that is recommended, which I've also talked about already before. For insects, the mounting medium of choice is the mounting medium called Euporol. I have it over here, written over here on the how it's spelled. Look here. Euporol mounting medium. That mounting medium is a standard which is used by entomologists. These are people who are basically preparing insects, because it has a so-called a clearing activity. This basically means that after a couple of days, or maybe after a week, the insect does not become as dark anymore. It starts to brighten up. So it has this built-in clearing activity, and there is a second advantage of using Euporol, and that is that what you do is you put the insect into alcohol to dehydrate it, to remove the water, and to kind of disinfect and make sure that it's free of bacteria, because the bacteria which are left on the insect, and if there's some water left, then the bacteria can actually break down and decompose the insect, and then you've got bubbles forming, decomposition gases forming. You don't want that, so you want to remove the water and disinfect the insect, and then you can take it directly from the alcohol and transfer it into the Euporol mounting medium, and the Euporol is able to handle the alcohol. So I'm saying this because not every mounting medium is able to do that. So in that sense, it's quite easy, how do you say, to prepare those specimens. So Euporol is the mounting medium of choice for insects. But what I'm going to do is I'm going to go back again here. Okay. So this is kind of the thing here. And what I'm going to do now is after aphid, I'm just going to work myself kind of through all of the slides that I have over here. And then we're going to see how far I'm able to go. Okay, but maybe we're not going to talk about the insects first. I want to get the non-insects out of the way first, which are the ticks. I do have a couple of tick slides here as well that I would like to show you. Okay, and some mites, simply to show you a little bit, because the mites and the ticks are indeed related. Simply for the sake of completion, because I made those. You might kind of wonder where I got those, not the Varroa one, the ticks. Where did I get those ticks from? Well, every time when I got bitten by a tick, not a good thing to happen, but I pulled it out of my skin and then I made a slide with the tick. Again, ticks are not nice things, because they do carry diseases. Okay, so that's something that you always want to be a little bit aware of. And let's remove the aphid. Let's just get the ticks out of the way, because I just wanted to show you a little bit the number of legs, to make an important difference between insects and and ticks. And so I have to find it first. Here it is. Okay, here. Okay, so that is the varroa mite. I've also made videos of this already. That's a very problematic one, especially for bees, because it's a parasite of bees. And yeah, if we look at it, for example, then you can actually see that you have got 1, 2, 3, 4 pairs of legs. So basically there are 8 legs. Yeah, just like the spiders have 8 legs, those mites and ticks also have 8 legs, and that's an important distinguishing criterion between insects and spiders and ticks, and of course no wings present either. Okay, so that is essentially the mite. I've made videos of this already before. Maybe you already feel a little bit bored about it because I've showed it to you so often. Okay, but now let's have a look at a couple of ticks here that I prepared as well. Okay, that's one of those ticks that I pulled out of my. I have to find it again. See, I don't even know where it is on the slide now. Just a second. Let me take it out again. It's over here on this side. I was looking on the wrong side. Somewhere over here it should be. Here it is. Okay, yeah. So that is a tick. And let's go up again with the magnification a little bit. I mean, to a certain extent, I do have to say, it does look a little bit kind of beautiful if you look at it like this. But then again, it's not one that you actually want to be bitten by, right? What we have over here is, again, you can see this leg is broken off. We've got 1, 2, 3, 4 pairs of legs, and also the proboscis, the little trunk which is used for sucking blood, was broken off here. Yeah, but and another no wings, as I told you. And another important thing about how spiders and insects are different, or ticks and insects are different, is the number of body parts. Because what we have here is that we have over here a so-called a cephalothorax, which is a head and a chest fused together in a large abdomen. So there are actually only 2 body parts here. And what you see here in the back, here, these circles that you see over here on the side, this is basically how the animal breathes. So that's basically an opening here, and this is so-called there is a network of trachea, which of tubes going inwards, which is used for breathing. So you have over here and over here openings on the side of the body, for breathing. Yeah. So, and you can see already here, I mounted this one in U-Pearl mounting medium, and it's pretty transparent. Okay. So you see that the mounting medium is pretty important here. So I've got another tick over here. Again, let's get the ticks out of the way because they are not insects. Yeah. You see they have different sizes. Yeah. Yeah. And over here, it also seems to be missing the place. The mouth parts seem to be missing. Maybe they got broken off as I pulled the tick out of my skin. Yeah. This was already, it's a pretty big one already, but you can also see that there are 8 legs. Yeah. So this is kind of a something I just wanted to show you. I every now and then again skip over to the questions. How do compound eyes work? Well, basically, it's like this, that compound eyes, ticks don't have them, but maybe I'm going to be able to show you a better one later on. They have they're pretty complex structures, but what they have is these are individual light-sensitive elements that are arranged in a pattern. And at the bottom you have, of these so-called ommatidia, you have light-sensitive cells. Right, so the insect will get a very almost a 360-degree view, but not as clear as our humans obviously have. But for that reason, they're able only to see basic shapes, but they're able to get a very good coverage. So this means they can go basically 360 degrees around. That's why it's kind of difficult to catch insects, because they're actually seeing what comes from behind. Yeah. So, would it be okay to use regular ethyl alcohol to preserve insects? Yes, because that's what I'm using. Okay, regular ethyl alcohol can be used, and that's basically what I've been using. It's 97% or so. It's fairly cheap to obtain. Yeah, and there's another comment further up. Crispy B, I do the same thing with mosquitoes, Gelsen. If one of them comes into my home, they get preserved in alcohol. Yes, yep. Especially during summertime, there are plenty of those. I've got plenty of mosquitoes here as well. Okay, so I just also wanted to show you now. Yeah, let's move on. I'm still with my homemade slides here. This one over here is a fairly large one. I wonder if you're able to recognize this one. Okay, let's find it again. Yeah, let's remove the... How many of you know what? Yeah, this is a pretty bad preparation because because of its thickness, the mounting medium already, yeah, kind of started to form bubbles and to separate. Maybe you can see that a little bit here. I wonder if you know what this one is. Completely harmless. Okay, it's a silverfish. Yes, exactly. Yeah, yeah, yeah. So that's and again, silverfish. They yeah, common in many households. As a matter of fact, they keep the place clean because they eat. Yeah. Yeah, and they eat the dust and dirt. And again, over here, if you zoom in, let's put this one in again. You can see that there is, yeah, we've got those eyes here. Usually eyes are very pigmented, very dark, therefore easy to see. But then maybe it's a little more difficult to see the compound eyes here. Yeah. It's a little bit more difficult to see. Yeah, but these are silverfish. Yeah. Yeah, so the comment here is they look pretty different when mounted on a slide. Yes, again, here this, yeah, the mounting medium makes a huge difference, but this one has already been preserved on the slide for quite some time, several months. Actually, it's a very sloppily made, yeah, specimen. So I'm a little bit surprised myself that it got preserved that well, yeah, because it's fairly thick, yeah, as you can see here. Yeah. So, so I'm still with my own slides. So I've got mosquito, lots of mosquitoes. What else do we have here? What is this here? June 11th, 2019, not properly labeled. So it's okay. Yeah, another tick. Yeah, I got plenty of them. Yeah, here you can actually see the mouth parts much better, right? And one thing that I've also seen once, and unfortunately I could not find the slide again, is I actually did find a tick with only 6 legs. This is kind of strange, I was wondering. Actually not. So this one has 8 legs, right? 1, 2, 3, 4 pairs of legs again here. Yeah, that's the opening of, yeah, for the digestive system. And this one over here, the 2 things here on the side are for breathing. The mouth parts look pretty scary. Now the one with the 6 legs these are also ticks and they're larvae. They're not adults yet, so this can be, don't confuse those with insects, yeah, because yeah, they actually only have 6 legs when they are immature and when they're larvae. Look at the mouth parts, pretty scary looking, right? Yeah. Again, you do not want to be bitten by those. They carry diseases. Borreliosis, which is a bacterial disease, also here in Europe, maybe also in the United States and other parts of the world. Besides borreliosis, we also got a virus infection called, which causes meningitis, which is caused by a virus. So they're not, you can get vaccinated against those, yeah? But yeah, you kind of want to try to avoid those guys here. Yeah? But I'm also kind of happy with this slide here that actually over the time it got pretty transparent. Yeah? So you're able to see quite many details and with minimum preparation. Dropped it into alcohol for a day or 2 and then transferred it into the mounting medium. Yeah. Okay. So I'm going to go through a couple of questions. Do most insects have compound eyes? I mean, I would almost, you see, I would almost say all insects have compound eyes, but then again, you in biology, you always have to be very careful when you say all or none, because if you look hard enough, you're probably going to find some exceptions, right? But generally, one of the characteristics of insects is that they have compound eyes. Yeah. But then again, maybe there are some around that maybe they live in complete darkness somewhere, and they've lost it. Yeah, because they don't need them. So I wouldn't be surprised if there are insects out there which do not. Yeah. Is a silverfish the same as a paperfish? I have not heard of the term paperfish yet. Okay, so I don't know about that. Yeah? Do you use both flat and weld slides? I will actually. Where are they? I don't have them here with me now. What I've got here these are so-called flat slides. Obviously, they're the cheap ones. They're completely flat, and then there are slides that have an indentation. Okay, I also use those slides, but I do not use them for making permanent mounts, simply because they're more expensive. But I do use weld slides if I want to look at live samples, and if I do not want the cover glass to crush the specimen. So it depends really on the specimen that I want to observe. Yeah. I remember when we looked at the compound eyes, they seemed to be made of circles. Do you think they might be the eye cells? These are called omatidia, the individual circles that you see. Actually, they're quite long. The compound eyes are long structures, right? Essentially what you see is only the outside part, right? And then these are kind of like cone-shaped, arranged next to each other. Okay, so what I'm going to do now is I'm going to start slowly moving over now to a couple of mosquito. Yeah, these are the mouth parts of the mosquito. Let's see if I'm able to find them as well. That's not, look at this. This is not such a nice specimen. I used a, what is this? Maybe, no, not a good, here it is. That's it. Oh gosh. Some time ago I actually isolated the mouth parts of the mosquito. You're the one that basically, yeah. Actually, I don't even know if this is the one, because actually there are several mouth parts that has, and only the central one is actually sharp and punctures the skin. I don't know about that. Yeah. But what you're able to see over here, that's also very typical. These are the scales. Yeah. So sometimes you actually see some of the scales. They look a little bit similar to the scales of a butterfly, but not identical. Yeah. Yeah. So the thing is, I'm reading again. By the way, where are the compound eyes on the tick right now? Well, ticks are not insects, so they do not have compound eyes. Okay, so they should actually have lens eyes. Yeah. And how come your insect slides are? How do compound eyes work? Well, basically, it's a different way. Let's put it. I don't have a model here right now, maybe we're gonna use some kind of, I don't know, a tube or something. No, what look at this. It's a syringe, okay? Just imagine the following. What a crazy comparison. Let's say that this is one of those elements of the compound eye. And basically what it is it's like a tube, right? And on the bottom of the tube there is a light sensitive cell. All right, and then the light comes in from the top, and then essentially, I don't know, like you've got many of those next to each other, arranged in a circle, and then basically the light is able to enter from the top, and then it's picked up and converted to nerve signal on the bottom. So it's not like, and each one of them has a very simple lens type structure in there, but it is not like this, that there is one lens like we have in our eye, but actually there's several elements that are kind of joined together, right? Yeah. So that's a little bit a very simple explanation. Yeah. How come your insects are so beautiful and not crushed by the flat slide? First of all, some of them are a little bit crushed, so that's one thing. And sometimes you might not see this. The specimen is so thick that the cover glass is not really flat, but it's kind of lifted up. Not ideal, but I would say that the commercial ones are probably even a little bit better. Yeah. From the previous videos, what is the volume of the auto pipette that you use? Is 2.5 microliters enough for protozoa? I think that's too small. 2.5 microliters. Just a second. Where is the pipette? Where did I put it? Just a second. Here it is. Okay. So the question was about this pipette here, which I can also recommend. And where is the desk view? Okay. And yeah, so and this one goes from 5 microliters to 50 microliters. You can adjust it here. And I've also got a bigger pipette, one that goes up to 1,000 microliters, but I found that to be a little bit too large. Okay, so I think with this one over here, I was doing fine. It's a question of preference. But 2.5 microliters is a little bit small. I mean, of course, you can try to take up protozoa this way. But what I usually do is if I want to suck up something, because I'm not using it for measuring, I just push it in slightly and then, yeah, take up whatever liquid there is in here. Yeah, I'm not using this for actually measuring out precise volumes anyway. Yeah. So I noticed that the UPRO preparations have no air bubbles. Is this the reason for this skillful handling, or can UPRO help a little? So first of all, the so-called air bubbles, they can form for several reasons. Air bubbles can form, especially if the mounting medium starts to shrink a lot. Nail polish does that. So that causes air bubbles. And another reason why there can be air bubbles is because when there is decomposition going on, when bacteria start to break down the insect, then gases like carbon dioxide and so on, methane maybe, can form, and they form bubbles. Not good. And this can actually cause the cover glass to lift away and lift off from the specimen. So the UPRO, because it is contains a solvent, it kills off all the bacteria. Yeah, so I would say that this is one of the reasons as well. Yeah, are ticks arthropods? Yes, ticks and insects are arthropods. Okay, so what I'm going to do now is because I spent a little bit more time already here than I wanted to spend. So these were my own slides now, but now I'm going to move over. I just want to work myself through those slides here, the commercial ones. I just want to show you the box here. This is the box which I bought. And in one of the previous videos, a couple of months ago, I actually looked at, I unpacked it and I actually looked at a few of those slides here, but not so many of the insect slides. Okay, so you see that the insect slides over here, I've taken out. We're over here all together, and I've put them now here on my table, and I'm just going to look at them one by one now, and just to have a little observation session here. But this one over here, I think I have shown you before. It says here ant. Okay, so, and just as an explanation here, when it says here WM, it means whole mount. Yeah, so most of them are so-called whole mounts. Basically, they just took the whole specimen, the whole insect, the whole leg of the insect, and simply mounted it. Yeah, and yeah, that's basically it. I think just looking at the color of the mounting medium, it is slightly yellowish. Okay, and this could be a sign that they also used UPRO, because UPRO is slightly yellowish. Okay, so that's simply something I just want to just over here. If you look at those here, this mosquito wing that I made, it looks, I don't know if you're able to see that, it does not look yellowish, and I used UKID mounting medium, right? So just by looking at the color, you can already kind of guess maybe what they used. But let's look at the ant first. I need to always switch over again, okay? And here we go. Here it is. Let's always start at the lowest magnification here. Yep. Now, unfortunately, it does not tell me which species it is. And what we have here as well is 1.5 ants. And then, a little bit dirty in the back. But we see over here the mandibles. Yeah, that's basically the mouth parts. That's the head of another ant. Yeah. Many of the legs are broken off, but what we're able to see here is the typical, the 3 typical body parts. Yeah, basically head, thorax, with the legs, and then the abdomen. Yeah, so insects generally have those 3 body parts. Yeah, and of course the antenna. Yeah, and some of the legs unfortunately broken off. Yeah. Yeah. So I never knew insects were arthropods. Are you saying that most insects have 8 legs? No, insects have 6 legs. The spiders and the ticks and the mites, they're kind of related. They all belong to the so-called arachnids, to the spider-like animals, and they have 8 legs. Okay. Arthropods, for example, also crabs belong to the arthropods. Millipedes, centipedes belong to arthropods. They have, again, different number of eggs. Yeah. So you see, the arthropods is like a big category, right? So, for example, I've showed you some time ago those ostracods, the water crustaceans, they are arthropods as well. Yeah. So that's the thing here. So that is basically the ant, and as we can see here, yeah. Maybe not such a nicely prepared specimen. Look at the legs. Yeah, I think still interesting to look at, right? But it's kind of, yeah. Over here, this might also be an interesting thing here. You can see that actually the abdomen, which is actually fairly large, over here is ripped open. Now this actually happens when you compress the insect. So it could be that they treated the insect in potassium hydroxide. That's one possibility, because potassium hydroxide makes the insect very soft and also transparent. And then when you squeeze it, then essentially it can break open and the contents kind of spills out. And so this is simply explains why it's kind of torn and ripped open here, right? Yeah. So that's the one. And this is actually one that I already showed you some time ago. So I will, you know what, I'm just going to stay with bright field, I guess. Yeah. I'm not going to bother about the filter, the polarization filter. Yeah. Because if I just stay with bright field, then that's actually the way that you're also going to see it in your microscope. So the next one is, it says here, what? Locust. Testes. Oh, okay. The testes, TS means transversal section. Never looked at this before. Okay. Okay. Okay, that's a histological specimen here. Yeah. Yeah, these are basically the cross section of the testicles of a locust. Okay, well now we know it. I think not so interesting now. It's a histological section. I don't know if we're able to see the gametes here. I know too little about this, about the insect. Could be. I don't know. Did they? What are we able to see here? How did they stain that? There's simply too, unfortunately. Could this be chromosomes? If this, then this would be again interesting. I don't know. No, let's switch up. Let's go to 60 times. I don't know. This would be actually quite interesting again. This is clearly a stained sample. Let's make it a little bit brighter here. And I need to focus. This looks, yeah, this is most likely. This could be indeed. That's maybe the reason why they included those. What I would guess is that those things, that's the nucleus definitely, and those little structures in there could be chromosomes. Because of course, as the gametes are produced, the cells divide. So I can imagine that basically, here again it looks quite different again. I wonder if these are already the finished gametes. Well, in any case, that's more a histological cross section, yeah. But I think we're just gonna go back to the whole mounts that we had before. Okay, again. So let's put this away. It's more histological sample. 86, 87, 88, 85 mosquito larva. Okay, that is now a larva, and this one is something that some of you might have already also found in water. I think they look quite nice when you are still alive because they're moving around. Okay. Okay, yeah, that's the larva of a mosquito. And here on the back, this structure, what you see over here, That is the breathing organ over here. So basically what it does is they swim to the surface of the water, they stick out the back part here for breathing. Yeah? And then the rest of the body here is essentially submerged in water. Yeah, yeah, so that is, honestly, I think that, yeah, when they're alive, they look a little more interesting. Yeah. Let's turn on the, here again a little bit. Okay, well, so that is the larva of a mosquito. That's also one of the characteristics of insects. This is obviously that out of the eggs that are laid, a larva hatches. And then it undergoes metamorphosis, and then you have the adult insect. So let me quickly go through over here. What are those elongated structures? I don't know now what you are exactly referring to. Yeah. A live mosquito larvae scared the heck out of me when I was viewing a drop of pond water during my microscope. Yep. Yeah. So that is yeah. Okay, let's go to the next one. So also maybe not the best thing to, or the easiest to mount, obviously. So the butterfly leg hole mount. Okay, so the next one is the leg of a butterfly. I always kind of regret a little bit that they do not give us a little bit more information about which type of butterfly it is and so on. And I have to find it because I don't have no idea. Oh, here it is. Huh. This is pretty transparent. Let's do it like this a little bit. Let's go bright field again. Yeah, and you see the slide's a little bit dirty. Yeah, so this is pretty transparent again, but we're at least able to see the claws here. Okay, which the butterfly uses for holding. So that's the, yeah, and in here you see the joint goes around here at the corner. Yeah, and then on the other side again. It kind of, normally the legs of insects, they look a little bit darker, and normally you can also see the muscles inside them, but this one seems to be quite transparent and pretty pressed flat. Yeah. So I don't know, it almost looks a little bit like it, yeah, like it's pressed flat. Yeah. Okay, this one I don't consider such a great specimen. Yeah, because, yeah, let's go up a little bit with the magnification, a little bit more. Let's get the arrow out of the way here. Yeah, I've seen more interesting yeah, insect legs. And it does seem to be quite empty. I mean, so maybe during the preparation they have removed the contents of the leg. Yeah. Yeah. So I'm just checking here again. Maybe yeah. Greetings from from all over the world. Still comments. Okay, no questions for me. Okay. So yeah. So these were basically the mosquito legs. So I still have quite a few to go. A fly antenna, hole mount. Let's have a look at this. Okay, so again I have to find it. Ha! Is this it? Come on, nah, I don't know. That's interesting still. Ah, that's the place basically where it was, yeah. Flies, of course, they do not have very large antenna, because otherwise, this is basically the place where it was joined. And I mean, I cannot talk about flies so much, but I guess here it's the same, especially some insects, also moths, for example. They have extremely large branched antennae, and they are used for picking up chemicals, obviously, and also so-called pheromones. These are chemicals that are produced by the insects for mating, so they're able to smell, so to say, each other sometimes over long distances. So it's important for reproduction. And what I read somewhere is that in some insects, those antennae are able to pick up. They're so sensitive that they're even able to pick up individual molecules of those chemicals. Yeah. So, but this kind of explains a little bit here. Yeah, I guess maybe it's similar with flies as well. Yeah. So there's a question, interesting one here. I'm sorry if I misunderstood, but why whole mount when they only show a part of an insect? That's a good question. The question is you call it a whole mount, WM, but you only basically show an antenna. The reason is it's still considered a whole mount because we take the whole part and mount it. We do not cut it. Okay. If the people who made the slides were to cut the specimen, then they would call it longitudinal section or cross section. But then it's actually sliced. But in a whole mount, you do not slice it. Yeah. So even if it's only part of an insect, if it is not cut into small sections, they still call it a whole mount. Okay. This is kind of the idea here. Yeah. Our woolly bears are they're male and females. I think woolly bears, are these type of butterflies. Yeah. Generally, what we because I know that sometimes I would say, let's put it this way: with those animals, in insects, you have all sorts of interesting combinations. Of course, you've got male and females, but in some insects, they are also able to reproduce parthenogenetically. This basically means that those insects are able to reproduce out of so-called unfertilized egg cells. Yeah? It's a form of asexual reproduction, which is also possible and which increases very rapidly the number of individuals. Yeah. So honeybee antenna, whole mount. Okay, there is a honeybee antenna here. Let me have a look at this. Where is the antenna? Here it is. Okay. Yeah. I like this one a little bit more. The contrast is a little bit better. Yeah. And also, of course, here, what we see, very typical of arthropods, what they have is arthropods means they have jointed legs. And of course, even though we're looking at an antenna here, we see that there are different segments that are basically jointed. Okay. Yeah. What we have over here is on the outside, this is known as the so-called the exoskeleton. It's made of a substance called chitin. And essentially all of the soft parts like muscles and blood vessels, all of the soft parts are on the inside. Yeah? So that's why insects are basically have this protective, you do not say shell, because shells per definition contain calcium carbonate. Yeah? But in insects and arthropods like mites and so on, we refer to this as an exoskeleton. Okay, so let's go up a little bit with the... Here, and here we can also see how the different segments are kind of joined together. So there's some other question. Hello, I'm new to microscopy here, and I've been watching your videos to get the black background in some of your pictures. Is it a separate dark field microscope or different lens or something different? Well, what you can... I do have a separate, you can easily convert a bright field microscope into dark field. But what I've got over here is I do have a dark field only for the low magnification. Yeah. So that is kind of dark field. See, Yeah, it's not working so well here. This is actually my phase contrast filter, which I'm abusing also for dark field. Okay, but so I'm not using this very often. But what you can do is you can essentially easily make your own dark field filter. But what I have got is on this microscope I can improvise a little bit dark field, but actually it's the filter for phase contrast. Yeah. Are scales cells? Strictly speaking, they are made of protein. Cells produce them, but they on them, to my knowledge, on their own are not cells. Okay, so this is a little bit like hair, hair or fingernails are proteins made of proteins, but they themselves do not contain cells. Okay, so this is kind of they're produced inside cells, and then the protein is transported outwards. Okay, so let's move on a little bit because I do want to go through all of those slides here. Again, okay, here we go. A mosquito wing. Let's have a look here. So I always have to of course look for the specimen a little bit. This is dirt. The slides are not so nice. Otherwise, I don't know what this is. Here we go. Yeah. Here we go. And yeah. So what we see here, of course, also the scales. Yeah. So yes. First of all, at the beginning of the video, I said that insects have 2 pairs of wings. Now, mosquitoes actually only have 1 fully developed pair of wings. The second pair of wings is highly reduced so that they're not even wings anymore. So there are basically in the insects, you have the so-called the dipteras, which have 2 pairs of wings, and the dipteras, they have. The Hymenopterus, they have 2 pairs of wings, and the Dipterus, they have 1 pair of wings. That's the way it goes. Doesn't matter. Essentially, the number of wings pairs is also one way how you can distinguish them. Also interesting, I find is why in the world do so many insects have hair or scales on them? Here you see the individual scales, but if you zoom in further, you can see that the actual surface itself is also covered by tiny little hair. And I guess this must have to do something with the aerodynamics. Because the wings are never, I've never seen smooth wings. They always contain scales or hair or whatever. On the, what you see over here, these here are vessels that transport blood, but at the same time they are also used for obviously giving structure and strength to the wing. Okay, so what's the function of the scales? Yeah, that's the question here. I also ask myself the same question. Of course, there could be maybe some sensory, it could be sensory cells connected to them to sense maybe the airflow. But again, I think this has to do probably something with the aerodynamics. Yeah, it increases the resistance, and maybe this is maybe by this way they're able to move a little bit more air when they're beating the wings. Yeah, I speculate. So, locust wing. So that is a mosquito wing. Actually, let me. This is the commercial mosquito wing. Look, I've got over here also a mosquito wing that I made some time ago, back in 2021, August 2021. I always put the date on it. And this is not mounted, this is in UPROL, I think. This is mounted in UKIT, which is a different mounting medium. Again, same problem, I have to find it. Also, this slide has collected a little bit of dirt. Let me quickly check. I have no idea. Just a second. Nah, it's way too dark. So here we go. Look. I used a round cover glass. Ah, here it is. Actually, you know what? This is, where's my, here it is. This is almost a little bit of an embarrassment. I'm gonna clean the slide now. Just going to dry wipe it carefully. It's a little bit better. So that's basically, this was now my specimen here. The wing is also a little bit torn, but here we can actually see the scales a little bit better. So what are the extra pairs of wings for? Is it if some have one, why do they need two? That's an interesting one. I basically, why have two pairs of wings if one pair is enough? Okay. The answer is, I'm going to give you a slightly philosophical answer. Why do we humans only have two eyes if two more eyes in the back would have an advantage that we could also see backwards? And the reason why is because it works the way it is. Okay. And essentially some insects have two pairs of wings, they're able to fly. Some have only one pair of wings and they're able to fly. You can solve the problem of flying in different ways, and as long as it works, it works. So there is sometimes not necessarily always an advantage or a disadvantage in biology, but sometimes there are multiple ways of how you can solve the same problem. To give you maybe a little bit more of a scientific answer is it could indeed make a difference concerning mosquitoes are much smaller than, for example, bees are. Therefore, the wing area that they need is enough if it's smaller. Okay, that's why they maybe have slender and narrow wings. It might depend a little bit on the flying speed, on the flying duration. So it could be that they're somehow optimized this way. So essentially, what I just want to say is there could be other factors that kind of influence that to optimize it. And indeed, bees are significantly heavier. They're larger and they need more wing area. So I assume that this is the reason. Now you could, of course, make the argument, well, could you then not just make one pair of wings which is larger? Yeah, I get the point. All I can say is it works the way it is. And if you look into nature, you see there is such a big diversity of living things. And sometimes there are different ways how the same problem can be solved. Yeah? And as long as it works. I think it's an interesting question, though. Yeah? Yeah, so... okay. Could you do a lens resolution showcase? I'm not quite sure what you mean. Basically, do you mean that I go up all the way with the magnification? Yeah. I'm a little bit confused by the question. Okay. So let's have a look at the locust wing, and you know what? I'm gonna wipe them. A lens resolution, maybe. Either compare different lenses, how they resolve. Yeah, the wing of a locust. Interesting. Fewer scales still here. Yeah, and here also. I don't know. Look, this wing seems to be kind of torn. It kind of makes sense as well, maybe because it's such a big wing that you cannot fit it on a microscope slide. Yeah, so of course you have to kind of separate it a little bit. Yeah. Yeah, but here again the vessels, and let's go up. I mean, what I could try to do is I could actually try to go up all the way as far as I can go, maybe to 60x, and let's have a look at I don't know. Let's have a look at this structure over here, how this resolves. I have to make sure that I'm using also the correct filter here. It's bright fields, and now I'm at 60x. And generally, what is the case is that, of course, as the magnification goes up, the lower the depth of field. That means that some parts in the background become blurry. So you have to kind of refocus this, and then those parts become in focus, and then the other parts become blurry. So that's essentially not a resolution issue, but a depth of field issue. So it might be also something that you might want to consider when choosing a magnification. So the question is, to know what resolution your lenses have. Let's put it this way. I could do that, but I would say that probably those specimens here are not the best ones because they're fairly thick. To do a lens resolution test, normally what you do is you use the shells of diatoms because they have very fine structures. And also another thing that I just want to say is that because I'm recording this on a camera and then it goes over YouTube and there's some compression going on, the clarity might not be the same as when looking directly through the objectives. But generally speaking, the further you go up with the magnification, the more blurry the image appears. And of course, you have on top here also the numerical aperture, so it is theoretically possible also to calculate the resolution. So, but I think thank you for the for the comment. But for doing a proper resolution comparison, I'd have to use different specimens. But I'm going to think about this maybe for an upcoming live stream. So what are those vessels for? Well, the vessels, which basically, let's go down a little bit. Insects, of course, they also have a circulatory system which transports nutrients and, of course, nutrients and oxygen, right? Because if they're living cells, then they need to be supplied as well. I could imagine, do you see this here, this very dark thing here? Okay. This is usually an indicator that there is air enclosed. Usually you see because of the large difference in refractive index between air and the surrounding medium, therefore usually air enclosures appear to be quite dark. And this kind of also shows that the inside of this vessel must be hollow. Otherwise there wouldn't be any possibility for air to be in there, right? So this kind of air enclosure here, or this dark area here, even though it doesn't look so nice, is actually an interesting sign which tells you a little bit about how those vessels are actually constructed, so that they are actually hollow. This is actually also a sign that sometimes that's also one of the reasons why you want to submerge your specimen first into some alcohol to remove any water, also to drive any air out, to make sure that then later on, when you mount it, that everything is properly filled up with mounting medium. Those little thorns here, what are those little protrusions on the vessels? Again, that's something that you find very often in insects and arthropods in general, are those little protrusions, that hair, little scales and so on. Also over here on the wing of the locust, if you look very carefully, you can actually see that on the wing surface itself, you can also see those tiny little hair. So there is an interesting one here by Marcel. The size of the insect matters in relation to the number of wings. In general, you might say 4 wings create more lift and stability. Yes. At the expense of maneuverability. Mosquitoes have to fly quickly and rapidly around and to try to fly away quickly when you try to hit them. And therefore they need to be very maneuverable. I'm satisfied with 4 times the 4 times objective as 0.1 numerical aperture. Yes. Yep. And hi Oliver, is it possible to make a permanent mount of vitamin C crystals? I have a slide that I made a few days ago and they still look very nice. Thanks. Yes, it is possible to make a permanent mount of a vitamin C crystal, but be careful. You have to use a mounting medium which is not water soluble. So that's very important. My suggestion is that you put a cover glass. If you have I don't know, I'm going to switch over now to the desk. I know it's a little bit of an off-topic question, but it's fine. Okay. I don't have those with me right now, but what you do is if you've got a specimen here, like a crystal, and you see you want to protect it somehow. So what I suggest is that you get yourself large cover glasses. There are actually large ones, and then you can actually try to tape them down on the side. Okay, then basically it's a so-called a dry mount, and then it's protected. And if you add a mounting medium, you have to really make sure, and you have to experiment a little bit, which one actually will not dissolve the crystal structure. Okay, so that is a little bit the issue here. And I know that vitamin C is dissolves a little bit in alcohol as well, so you have to be a little bit careful. I would experiment. Okay. But the first thing that I would try is to preserve them a little bit using a dry mount. And I also have to say something: vitamin C likes to oxidize in air. So actually putting a cover glass on top of it or making a permanent mount might also preserve it better. Okay. So hey, I've recently bought a microscope for brine shrimp. Do you have any advice for viewing brine shrimp under a microscope, especially for larvae? I would like to refer you to one of my previous videos where I put those so-called sea monkeys, which are brine shrimp eggs, and sea monkeys under the microscope. Okay, in this case, I do recommend that you get those slides that have an indentation. Let me quickly, I don't know if I have, just a second, give me a second. I don't have them here now. I don't know where I put them. But there are slides which have a so-called a concave indentation, and then basically otherwise there's a little bit of danger that the cover glass is going to crush them. Yeah. So let's move on. Honeybee wing. Okay, again wing. I think we've got the wing section now here. So let's put a honeybee wing. Yes, that's a classic. Yeah. So let's go down with the magnification here. Honeybees have two wings. So this is the front wing, the main wing, so to say. Yeah. Here, the vessel is very clearly visible. Yeah. So nothing so far. And the next one is the wing of a dragonfly. Where is this here? Look, that's kind of obvious. It's square. Why? Because, of course, the dragonfly wing is so large that you're not able to fit it on a microscope slide. Therefore, they had to, of course, cut it apart. Look, here, these vessels are interesting. Look, that's also quite interesting to see. Could this be some kind of strengthening rings, maybe? Let's go up. I'm speculating a little bit. Let me put this here away. That could be. Okay, look at this. Again, that's a speculation from my side. That could be a vessel over here, and those things over here that seem to be kind of like we also have it here. Yeah, here, like going across like rings that kind of stabilize the structure. Okay, it reminds me a little bit of the so-called xylem vessels, where we have a similar thing. Yeah. Yep. And here, again, one of those veins here. So that is basically a dragonfly wing, and here that's again the honeybee. Yeah. I mean, if you think that they kind of do look similar, right, then that is indeed what we call in biology homology. They have, of course, they look similar, right? Look, that's also nice. You occasionally find this even in honey. Those little strange structures are also basically some hair that you sometimes find on the bee itself. Yeah. So I found it looked like a little bit like tiny branches here, but I found them also in honey. Yeah. So these seem to be also kind of hair that are found on the bee's body itself. Yeah. But they are now here also on the wing. Yeah. So I quickly check the. Yeah. So far I did not see any questions for me. Do all wings have those small hair? Well, honestly, I've looked at many insects, and all of them basically had more or less of these hair. Yeah. So I think they must fulfill some kind of an important function, otherwise they wouldn't be there, obviously. So the next one, butterfly wing. We were talking about hair. The butterfly wing, of course, we know are rather extreme. We do not only have hair here, but look at this scales. And those scales, are fairly large. Here, that's actually a nice one over here. And those scales can sometimes also be found in dust. So every now and then, when people or in water samples, they observe something and they find a scale like this, kind of wonder what could this be. Yeah, and it's in many cases it's a scale of a butterfly wing. Yeah. So. Yes. Here, this is the sample that I really wanted to talk about as well. And I made a video of this as well. Look, this is something where I really need to go back a little bit in time. It's this mystery object here. And this here, what in the world could this be? Okay, look at this. It looks really strange. And I found this also in dust, in house dust. In my own, So, and I was kind of wondering, what in the world is that? And I found this several years ago, And also in this sample here, it's a commercial sample as well. Some kind of interesting here, but what could this be? And several years ago, I did post a question in a microscopy forum, and I even got an answer. And as a matter of fact, these are the so-called the setae, which are the hair of the larva of a carpet beetle. A carpet beetle is an insect, obviously, and it has a larva, and those larvae are very hairy. And this is one of their hairs. And the question is what in the world does this have to do on a sample of a butterfly wing? Yeah. And the thing is evidently the company who made and prepared those specimens, obviously they kept their butterflies, and it must have been contaminated by one of those larvae, because they eat insects as well, right? So it was quite fascinating. Yeah, but these here again that you see over here, those setae are essentially the hair of the larva of a carpet beetle or museum beetle. Okay. I made some videos about this as well because I was really surprised. Yeah, and I would have never identified this myself, but I got a hint by some people. And if you actually go into Google and if you type this in, they have a very nice term. If you speak German, then try to Google the term Pfeilhaare, arrow hair, if you were to translate this. The Pfeilhaare. And then you're going to find in Google, you're going to find actually, those here as well. Yeah? So museum beetle or carpet beetle, it's called. Okay, yeah. So it's so actually what we have is we have two different insects here: the butterfly wing and those tiny here. As you can see, there are many more. Yeah. Look, all of those long structures here. Yeah. Pfeilhaar, yes. Pfeilhaar, yes. So that's if you Google this, then you should be able to find a couple of pictures of those. Yeah. I found this kind of fascinating because I did not expect this there, right? Okay. Oh gosh, now it's becoming boring. This is basically the wing of a of a housefly. Okay, you get the idea. Yeah, housefly wing. Yeah, I think we're just gonna move on. Pollen-carrying leg of a honeybee. Okay, and you know what? It's already over an hour. Let's just finish off the remaining slides. So this is now from look at this. That's a honeybee leg. That's a pretty huge one. Yeah, evidently, look here as well. You can see that the specimen is cracked open, which I do not consider such a big problem because it's actually a sign that it was compressed flat. Yeah, and you have to actually make sure it's kind of flat. Yeah, so those tears and rips in the specimen are just basically are preparation artifacts. Yeah, let's go up with the brightness a little bit. So that's one of those hind legs. Yeah, it's a huge specimen again. So looks quite fascinating. The complexity of nature. So complexity of nature. So let's move on again. So I'm just reading every now and then. If I'm pausing, then I'm just to let you know that I'm reading a little bit the comments here as well. Honeybee, foreleg, whole mount, another leg of a honeybee. Okay. Yeah. Also a nice claw here. Interesting still. Yeah, but I think all of those specimens, they look very, very thin, which is actually good. But this actually means that probably the contents has been removed during the preparation. Yeah, during the preparation of the specimen. Yeah. So. And here are the legs. First we had the wings, and now we were with the leg section, mosquito leg. Yep, leg of a mosquito. Let's have a look. Wow, that is a long one. I mean, if we know that mosquitoes have long legs. But I don't do it. Does this have a claw? I mean, mosquitoes are able to sit also on walls and everywhere. So they must have, there might be a tiny claw over here as well. Yeah. Yes, here it is. Yeah, you see, almost those, those two finger-like extensions here. Yeah. I would love one day to see an I cannot read this now, unfortunately, because there's this thing covering this. I would like to see an insect leg damaged by diatom earth. Will we see the scratch? You mean diatomaceous earth? I mean, I do have diatomaceous earth at home. So basically that... So, interesting. Basically the diatomaceous earth being in contact with the insect, you mean, because... yeah. Would you focus on the spike of the leg, the one just below the last leg? Which one? An insecticide. I see. I see. So basically, the diatomaceous earth. I don't know if the diatomaceous earth, which kills, which is very fine powder, which if it actually damages the exoskeleton, but it could be maybe that it's taken in or inhaled maybe by the insect, and maybe that this kills it. I don't know. Okay. Yeah, it's a bit further down. Yeah, there was some. Yeah. Silica. It's like tiny needles. Yes. I don't know. There was some kind of a spike or a spoke or something like this. I don't know. There's been a request that I focus in on something here on the leg. This is huge. This thing. Look at this. Lots of scales here. Okay, that's where the next leg segment joins in. Ah, yeah. Maybe is this the thing? Yeah. Yes, these were spike protrusions on the lower parts. I've seen it on a lot of butterflies and moths too. And you know what? I think I do have to show you. Then yeah, I've got also some other slides as well. Maybe one of my favorite ones. Ah, here, look at this. Maybe here is also a spike. But look how pointed it is. This is really, I think it is really amazing. Yeah, it's really, even at this high, I'm using my 60 times. It is really, really pointed. Yeah, quite interesting. Okay, you know what? Very quickly. I do want to also show you something. Just housefly leg. Yeah, here the leg. Yep. But here I am not happy with this one over here, because I think here the front part is actually missing, because the leg of a housefly is actually cut off. I think this is actually broken off. Yeah? So that's not the end, because a housefly leg also has those two claws. Yeah. Yeah. So. Ah, yeah. The advertising of diatomaceous earth says it damages the exoskeleton and the insect dries out. Ah, that's interesting. Didn't know that. Okay. I should zoom in on the mosquito proboscis. Okay, I don't know where it is now, unfortunately. Dragonfly leg. Go down a little bit. I've got now the whole table is full of slides, all completely jumbled around. So where is this here? This is the leg of a dragonfly. Ooh, that's a huge one. Okay. Oh yeah. Now, these are some gigantic claws here. Huh? That's interesting. Yeah. Okay, and mosquito honeybee antenna. We already talked about that. Mosquito leg. We talked about this, and there was one thing that I wanted to show you. Where is this? Just a second. I don't know if I can find it again. But that's the one over here. Just give me a second. Locust. If not, these are the mites here. Mosquito wing, mosquito legs, locust. There was actually somewhere. Nope. These are the mite, varroa tick. Okay, mosquito wing, mosquito leg. No. You know what I'm going to show you as a last sample? Just a second, I need to get it. Maybe one of the nicer ones. And I've got another slide box here. Number 15. Here it is. That's another slide. I've got the insect. I've got a second box with some insect slides, and that is also from a honeybee. Where is it? Look at this. I wonder if you're... That's the stinger of a honeybee. That's why honeybees hurt when they sting. Yeah, it's because of this stinger over here. And here in the back, somewhere, there must be, I think, the gland that produces the poison. I would say probably, probably this one over here. Yeah. And over here on the other side, you've got the stinger. Yeah. That's a pretty scary looking thing here. And if you look very carefully, then you're going to see that it's not smooth. But they're kind of tiny little hooks on here. And that is the reason why when a honeybee stings you, then the stinger actually stays in your skin. Yeah. And the question about the sharp spike on the leg, I don't know what the function of the sharp spike on the leg is. But if you really want to see insects with a lot of spikes on the leg, then I can... Where is this? I can recommend specimen number 10 here. This is a flea, a whole mount of a flea. That is another insect. And look at those spikes that we have here on those legs. Yeah. And here I can actually tell you what the function is. It is actually very important for jumping, because those fleas, which are able to jump very far, they will actually make contact with the ground with the whole lower leg part. So that means actually they will, those spikes will actually go into the ground, because it's not just jumping off with a foot, but it's actually putting the whole lower leg into the ground, and it's pushing itself off. Yeah, so they're kind of anchoring themselves into the ground to create a lot of friction, so that it's able to jump off. Yeah, you can actually have made slow motion videos. I've seen this somewhere on YouTube, which actually showed that. Yeah, so those basically are pretty important for creating that friction. Yeah, because when it jumps off, they will basically place the whole lower leg on the surface, and this causes the spikes actually to bury themselves into the surface, and then they can push off much faster. Yeah. So is this a commercial slide? Yes, it is. Yeah, the one with the stinger. As a matter of fact, most of the things that I showed you were commercial slides. Yeah, and this one over here is also a commercial slide, and which you can essentially buy. And that's the one I actually wanted to show you, and I forgot where I put it. I don't know where I put it. There was a slide with the stinger of a wasp, which I made, and I can't find it anymore, unfortunately, which is a pity. But yeah, it doesn't matter. You know what? If I find it, I'll show it to you. The wasp stinger is one that I got out of a dead wasp some time ago. I dissected a wasp and I actually pulled out the stinger, and I mounted that as well. And sloppy as I was, I didn't properly label the slide, so I don't know where I put it. But I just had it before I started the live stream. Yeah, yeah. So that is a little bit. Yeah, the thing. Where's the stinger here again? Here it is. Yeah, that's the bee stinger. Yeah. I wish I had the guts to watch insects under my microscope. As a matter of fact, I think that if you're specifically interested in insects, then I can highly recommend stereo microscopes, because they do not require a lot of preparation, and essentially you can put the specimen directly under the microscope. I don't know, maybe these are not the ones here. Varroa tick. Okay, tick. Yeah, mosquito wings, mosquito legs, mosquito. So many mosquitoes. Yeah. Yeah. But you know what, people? What time is it now? It's already 1 hour 24 anyway. Yeah. Last question here. How do you dissect specimens? Any special equipment? I can actually show you what I recommend. It's actually quite simple. The most important tools for dissection are... Where is this? Is I switch over, and that's essentially what I've used here. Yeah, very fine scissors, not a knife. Okay, scissors. Some people think of like a scalpel, like for the doctor uses. I don't consider them very useful because then you have to apply pressure, and no. This is basically really useful, and some kind of tweezers to hold it, or you can actually also hold it with your fingers. Yeah. So, but I would say that this is very useful here. Very finely pointed scissors, and you can basically cut open the insect, and that's what I've done. You can cut off the wings this way. Yeah. Of the insect and mount them. So this is essentially almost all that you need. Yeah. So that is where I'm just looking around for. So this is something I can recommend. For tools, I also recommend if you want to cut apart slightly larger objects, plant specimens or so. Of course, you might want to use slightly more solid scissors here. These are basically dissection kits, and if you go on Amazon and if you Google dissection kits, then you find they don't cost a lot, but usually in those dissection kits you find more tools than you actually need. So I would say that a tweezer and scissors like this are, I would say, and here it is. Those fine tweezers are also very useful. Fine tweezers and those scissors, and you basically are covered with most, I would say, with most of the things. So this is So you know what, people? I think I'm just going to leave it at that again. Just wanted to show you again a few of my slides. Hope that it was nevertheless interesting for you. I hope I was able to answer some parts. Okay. Okay, I see that there are a couple of questions here that I did not respond to yet. I often look at dead insects I find around the house. On my compound microscope, I have difficulty with lighting from. Could you suggest any lights? Well, generally, if you put the whole insect directly on a microscope slide, then it's too thick. Right, and then you're not able to. But what you can do in this case is you can place the insect. You put in the low power. You put the insect on a blank slide, and then you use a strong flashlight to observe it from the top. So that is also possible. If you've got a whole fly or whatever, any object, you put it directly on the slide here. Take a strong flashlight, and with a low power 4 times, you're actually surprised. You'll get a surprisingly good result. Could I enlarge the stinger? Okay, this also has been a final request. Let's go up all the way here. I need to, let's see how the stinger is. Let's remove the arrow. So 10 times, 20 times. You know what? I'm going to remove the filter again. This is now regular bright field again. You see now the little hooks in there. 40 times. In the center of the stinger, the canal for the poison. So now you know why. A bee stinger. If a bee stings you, the bee dies, right? Because it will rip out its abdomen and everything. 60 times. See, I think this is already a little bit too. Maybe the specimen is too thick already. So that's 40 times, 60 times. See, I'm not able to focus this because I'm kind of worried that I might be already touching the cover glass. So I'm not able to go higher than 40 times. 60 times I'm not able to get a sharp image. And even when I focus, I'm not getting a sharp image because I guess I'm already making contact with the cover glass because it's so thick. So that is the highest that I'm able to go, 40 times. Okay. So again, stinger of a bee. And was kind of, yep. Okay. Even with 40 times, it's I'm making already contact. So that's a difficult specimen. It's very thick, and the distance to the distance is so small that sometimes I'm actually making contact with the objective. Yeah. So. Yeah. So you know what, people? I think I'm going to call it quits for today. And unless there is something else here, okay? Yeah. Yep. So that's basically it. Yeah. So I think I'm going to leave it at that. I hope that you liked the live stream again. It's again, well, 1.5 hours, a little bit longer than I anticipated. But yeah, many of the specimens that we looked at today, I've also seen for the first time. I simply kind of kept them and reserved them for the live stream today. If you have any comments that you want, certain things that you want me to look at, I'm always happy for feedback and information. If it works out again next week, then I'll try to be again online. And then I'll think of another topic to cover. Yeah, I wish you all the best. Okay, happy microbe hunting as always, and hopefully see you again. Okay, bye-bye.